Okay. All right. Okay, everybody. Welcome to the Immuno webinar this week. It's really a great pleasure for me, both personally and scientifically, to introduce today's speaker, my friend and extraordinary immunologist, Professor Alexander Sasha Rudensky. Sasha is chair of the immunology program at the Sloan Kettering Institute, director of the Ludwig Center for Cancer Immunotherapy at the Memorial Sloan Kettering Cancer Center, holder of the Lloyd Old Chair for Clinical Investigation, and an investigator of the Howard Hughes Medical Institute. He is internationally recognized for his foundational contributions to our understanding of regulatory T cells and immunotolerance. In particular, his work established the central role of the transcription factor FOXP3 in the development and function of the regulatory T cells, transforming the fields of autoimmunity, inflammation, tumor immunology, and cancer immunotherapy. His achievements have been recognized with many of the most prestigious honors in biomedical science, including the Crawford Prize in Polyarthritis, the Wilczek Prize in Biomedical Science, the William Coley Award, the AAI Thermo Fisher Meritorious Career Award, and so on and so forth. He is an elected member of the U.S. National Academy of Science and the National Academy of Medicine, and a fellow of the American Academy of Arts and Science and the AACR Academy. Sasha, it's truly an honor to have you with us today. Welcome, and thank you very much for accepting our invitation.
Well, thank you, Gu and Bruno, for inviting me to this interstate, very special interstate, both within Brazil and within America at large, very special seminar series in immunology. Today, I will share with you recent work from the lab related to differentiation and function of regulatory T cells. But before I do that, I would introduce you to earlier work from the lab, highlighting the role of transcription factor FOXP3 in biology of the cells. And as Gu already said, now 25 years ago, FOXP3 gene was cloned as a gene whose mutations in mice and men were associated with severe, fatal autoimmune and inflammatory disorder. And this disorder is known in humans as IPEX. IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. And the similar fatal disease was found in spontaneous mouse mutant called scurvy. And this work was done by Fred Ramsdell's lab and Talal Shatila's lab, who essentially contemporaneously identified FOXP3 mutations in mice and men. Work from the lab showed that all the disease manifestations in FOXP3 deficient mice were due to deficiency in regulatory T cells. So this argument is based on the following observations that are summarized here. Work by former graduate student in the lab, Jason Fontenot, showed that FOXP3 is expressed in CD25 high and CD25 low Treg cells, but not in any other cell types. And that was done using first FOXP3 reporter mouse generated in the lab. This work also showed by Jason a requirement for cell-intrinsic FOXP3 expression for regulatory T cell differentiation in healthy mosaic mice, suggesting that FOXP3 does play a role of regulatory lineage-specifying transcription factor. Importantly, work from the lab showed that FOXP3 is dispensable for cell-intrinsic mechanisms of regulation of T cell tolerance and T cell responses, and that T cell restricted FOXP3 deficiency is indistinguishable. It's essentially a phenocopy of germline deficiency in this gene, and that neonatal transfer of regulatory T cells can rescue disease in FOXP3 deficient mice, and finally, that ectopic FOXP3 expression can confer suppressor function to activated T cells, and similar results were reported by Shimon Sakaguchi's lab and Fred Ramsdell's lab. So this work showed that congenital regulatory T cell deficiency resulted from FOXP3 mutation as fatal, and that Treg cells are essential for preventing, in early life, for preventing severe fatal autoimmune inflammatory disease. But we also demonstrated that this requirement for Treg cell function is a lifelong need for these guardians of immune homeostasis of the organism, and that was shown by John Kim when by generating mice harboring diphtheria toxin receptor, simian diphtheria toxin receptor under control of the endogenous FOXP3 locus. And the use of this mice showed that both in neonates and adults, depletion of Treg cells in perfectly healthy adult mice results in similarly devastating and even more rapidly progressing autoimmune inflammatory disease. The work that I didn't cite here, but it's important work nevertheless, by Takatoshi Chinen, showed that this inflammation and autoinflammation and autoimmunity was observed upon depletion of regulatory T cells was independent of commensal microbial community, suggesting that this function of Treg cells is to limit pathogenic self-reactivity that results likely from a self-MHC restricted recognition of T cells that emerged in vertebrates, in early vertebrates. All right, so analysis of mice deficient in Treg cells congenitally or subjected to ablation of Treg cells showed that the cells control all known types of inflammatory responses. So further evidence in support of the role of FOXP3 as regulatory T cell lineage specifying transcription factor came from our studies of mice harboring FOXP3 reporter null allele, and I would call them regulatory T cell wannabes, that receive all the signals that require for FOXP3 expression but lack FOXP3 expression, and mice that harbor functional FOXP3 reporter allele. Comparison of these mice showed that FOXP3 confers suppressor function and fitness to developing regulatory T cells, and also showed that transcriptional features of regulatory T cell wannabes and Treg cells are quite similar, although there are important differences, and I will highlight these differences and focus on this Treg wannabe versus functional Treg cell comparison later in my talk. Also, early work from the lab by Luke Williams, graduate student, showed that loss of FOXP3 expression in differentiated Treg cells results in a loss of function, and since this observation of Luke, now almost 20 years old, there is a sort of common notion that continuous maintenance of FOXP3 gene is required for Treg cell function, but there is an important new study that I'm about to highlight later in my talk, and I would like to revisit this notion later, so that it should be a more nuanced notion, I would say. All right, so with this in mind, the question, of course, was, are the regulatory T cells necessarily a dedicated cell lineage? Do they show stability in settings of inflammation that is known to promote effector cell function? So this question was addressed by Yuri Rubtsov together with Rachel Neese when they generated mice that enabled fate mapping, inducible fate mapping of regulatory T cells. This mice harbor CRE recombinase fused to estrogen receptor ligand binding domain, fused to GFP reporter, under control of FOXP3 locus and equipped with recombination reporter allele. This is safe harbor allele, ROSA 26, equipped with lock-stop locks cassette and YFP reporter, activation by tamoxifen of CRE recombinase, excises reporter, and tags the cells irreversibly with yellow fluorescent protein. So I will be using later in the talk this fate mapping trick and we'll talk about this more. So the studies showed that FOXP3 expression is stable in differentiated Treg cells, but not in newly generated Treg cells in physiologic and inflammatory settings. And the study also showed that regulatory T cells in adulthood are maintained largely through self-renewal, and that recent thymic immigrants, that continuous thymic output, makes very limited contribution to peripheral Treg cell pool in adult mice. All right. So while so far what I told you, this experiment showed that Treg cells are capable of preventing fatal inflammatory disease, this experiment didn't show whether Treg cells are capable of functioning and controlling already established severe inflammation. And the answers to this question were not clear in the literature. A number of publications suggested that Treg cells are disabled, or effector innate and adaptive immune cells become resistant to control afforded by regulatory T cells. Other studies suggested that Treg cells, to the opposite, are functional and perhaps gain in functionality in inflammatory settings. So there was no really clarity here. And with this, Wei Hu, former postdoctoral fellow in the lab, and Zhongmin Wang, former graduate student in the lab, decided to address this question in an experimental system that would have the following features. So they want an experimental system that would be characterized by systemic polytypic, meaning multifunctional inflammatory disease. So polytypic meaning type 1, type 2, and type 3 inflammation, type 3 inflammation mediated by Th17 cells. The system that would allow temporal control of regulatory T cell function in disease settings, and that would allow to avoid adoptive transfer of Treg cells because of the obvious confounders of adoptive transfer with uncertainty with migration numbers and state of cells. And the system that would allow extended longitudinal observation of mice. And thinking of an experimental system that would have this all four features, Wei and Zhongmin thought that actually FOXP3 deficiency itself offers such a system. With this in mind, they used mice generated in the lab by former postdoctoral fellow Yong Feng that harbor a reversible FOXP3 reporter null allele. We call this allele FOXP3 LSL, and that LSL stands for LOX, stop LOX. So this is the schematic of this allele, and I'm talking about this. The paper was published 4 years ago, 4.5 years ago, because I will be using this tool, genetic tool, in experiments I will describe later in my talk. So at the basal state, FOXP3 is not expressed because there is a LOXP side flanked stop cassette with Ty1.1 reporter embedded in the FOXP3 locus, and that followed by the functional FOXP3 gene with GFP reporter. So in mice harboring FOXP3 LSL allele, in male mice, that is, right, because FOXP3 is subject to X-linked X chromosome, random X chromosome inactivation. So in male mice harboring this allele, regulatory T cell wannabes are present, and they harbor Ty1.1 marker. And these mice are as sick as mice lacking FOXP3 gene, as I showed you before. FOXP3 is required for suppressor function of Treg cells and their fitness. So, however, after recombination that can be induced by 4-hydroxytamoxifen that is cleared after administration into mice within 48 hours, as we have found, one can activate CRE-ER encoded by CD4 locus, and that would induce recombination, excision of the stop cassette, and transition from Ty1.1 Treg wannabes to FOXP3 expressing cells, Treg cells. And that's the experiment, the way it was set up, outlined here. Then Wei and Zhongmin did wait for 2 weeks. At this time, mice become severely sick. They develop clinical signs of disease, and they administer once 4-hydroxytamoxifen, and they essentially convert Treg wannabes to FOXP3 expressing cells within 48-hour window. It's a single cohort of cells that is now acquiring FOXP3 expression. And then they monitor mice. And as you see here, the mice were rescued by installing FOXP3 expression in a cohort of regulatory T cell wannabes. And they were rescued for life, whereas mice that were genetic control mice died within expected time frame. All right, so analysis of this mice that I don't show because the data were, this was published some time ago, showed that all signs of immune activation of innate and adaptive immune cells, CD4, CD8 cells, B cells, antibody production, activation of macrophages, and most importantly, tissue inflammation was reversed in both neonates and adult mice upon a restoration of Treg cell function. In adult mice, we used heterozygote females that were treated in a certain way that, for the lack of time, I'm not going to describe this, but if you're curious, I would encourage you to have a look at this 2021 Nature Immunology paper. So together, the study showed that FOXP3 expression confers suppressor function, suppressor activity to non-functional Treg wannabes, and that a single cohort of Treg cells can persist and maintain long-term immune balance, as this paper also identified. Markers of putative resource Treg cells, cells that can long-term persist and give rise to terminally differentiated effector Treg cells. So the study also showed that Treg cells gain functionality in settings of established severe polytypic type 1, 2, and 3 inflammation, and that they are capable of reversal of systemic autoimmune inflammatory disease and tissue inflammation. All right, so the question is, how does FOXP3, which we believe is a lineage-specifying transcription factor, work? Known lineage-specifying transcription factors are thought to bind to closed chromatin, remodel chromatin, establish distinct set of cis-regulatory elements, and define gene expression program and functional program of differentiating cells. Unlike this archetypal, if you will, lineage-specifying transcription factor, FOXP3 works differently. We found in studies performed now years ago, at that time we used DNase hypersensitive site sequencing rather than attack sequencing. That was in pre-ataxic era, as well as chromatin immunoprecipitation and other readouts to show that FOXP3 binds to preformed, to open chromatin sites. And these open chromatin sites are occupied by transcription factors that Depay Rudra found in his study to be partners of FOXP3 in FOXP3 transcriptional complexes. Prominently, these are RANKS family members and ETS transcription factor family members. The other side of the other set of cis-regulatory elements, chromatin sites that FOXP3 binds to, are actually formed in response to signals that induce FOXP3 expression, most prominently T-cell receptor activation, and as well as IL-2 signaling. So these sites are occupied by, are remodeled and opened by likely AP1 members, transcription factor family members. And they're occupied by NFAT and AP1, and the sites are bound by FOXP3, which was shown to form complexes with these transcription factors. All right, so so then the work that more recent work that was performed by Joris van der Wieken, Ari Glasner, and Izong. Izong was computational biologist, postdoctoral fellow in the lab, is summarized on this in this panel. You can ignore the rest. By using mice with divergent, highly divergent genomes, that separated 1.5 million years ago in polymorphisms, and by using F1 mice where we can look at allelic imbalances and polymorphic sites, using this genetic approach with multiple omics readouts and reverse genetics, these three postdoctoral fellows were able to show that FOXP3 binding does not affect global FOXP3 dependent chromatin accessibility or gene expression, and that FOXP3 acts in a relay-like manner by modulating expression of a small number of direct targets. Some go up, some go down. Some of these factors are sequence-specific transcription factors. Others are signaling molecules that result in modification of activity of transcription factor, other transcription factors. So this allows for FOXP3 by tweaking expression of few direct targets to then modify expression of genes across the genome, and these are indirect targets of FOXP3. And what Joris and Ariel and E found that the majority of genes that are regulated by FOXP3 are regulated in indirect manner through this intermediaries, if you will. So this type of regulation raises the question of FOXP3-dependent gene and functional program, and specifically how vulnerable or resilient these programs must be in health and disease. So this question was addressed by Wei Hu in her recent studies. And the question that we had in mind was due to confounders of this previous work that I highlighted in the beginning of my lecture today. And that is this experiments by Luke Williams, where I told you that loss of FOXP3 in differentiated Treg cells results in a loss of function. So the design of this experiment was the following. At that time, we generated floxed allele of FOXP3. We isolated Treg cells harboring floxed FOXP3 allele. They were activated in vitro, and then using retrovirally expressed CRE recombinase, and this was transient expression of the CRE recombinase by hit and run retroviral vector, removed FOXP3, and then the cells were transferred adaptively into lymphopenic mice together with effector cells. And this experiment showed loss of function and assumption of effector pro-inflammatory function by this Treg cell subjected to loss of FOXP3. So the caveat of this experiment was that in order to remove FOXP3 gene, we needed to activate and drive the cells into very robust cycling. And after ablation, the cells were transferred into lymphopenic mice where they also underwent robust cycling. So then that left the question to us open whether FOXP3 is required really in vivo in differentiated Treg cells. And in order to address this question, Wei developed chemogenetic strategy for inducible FOXP3 protein degradation in vivo. And the strategy is based on the use of AID degron. AID stands for auxin-induced degradation or auxin-induced degron. So this is a peptide sequence that is recognized by TIR1, which is an adapter protein isolated from plants, that in plants is can be activated by auxin. Auxin is a plant hormone, but it's a simple chemical compound, it's iode acetic acid. Iode acetic acid binds to binding site in tier 1, and this binding site then allows for tier 1 to bind to this particular peptide sequence, degron, and recruit SCF ubiquitin ligase complex that would result in ubiquitination of targeted protein and its degradation, as shown here. Wei figured out the linker and equipped FOXP3 with ID degron that would allow its proteasomal degradation, and she optimized the system for in vivo because non-modified ID degron doesn't work in mice. It works in flies or in worms, but not in mice, as we found in a painful manner ourselves. So instead, we used mutant tier 1 that harbors mutation of the phenylalanine in position 74 in the binding site of iodacetic acid to glycine. So this mutant tier 1 was expressed in self-harbor locus, ROSA26, preceded by LOXP flank stop cassette, followed by mCherry reporter. So FOXP3 was engineered in the following manner. It was equipped with AID degron sequence, peptide sequence, to be recognized by tier 1, mutant tier 1, followed by FOXP3, followed by internal ribosome entry site and transcriptional ZS green reporter, followed by T2A, which is a self-cleaving peptide that was followed by CRE recombinase. So combining this ROSA26 tier 1, mutant tier 1 allele with FOXP3 AID allele enabled Wei by using now 5-phenyl iodacetic acid. So just note that phenyl group in this experiment moved from the binding site phenylalanine that was replaced by small amino acid glycine to iodacetic acid. So from receptor to ligand. And that allowed Wei, upon administration of 5-phenyl iodacetic acid, to degrade FOXP3 protein in vivo to completion within 6 hours. All right, so this is technical details of the system. Now, how Wei has used the system. So first, Wei compared consequences of ablation of Treg cells using diphtheria toxin that I described to you that results in death of mice within 2 weeks. To degradation of FOXP3 protein using this chemogenetic tool that I also described to you. And as you note here, then on day 14, there is a massive splenomegaly lymphoproliferation in mice about to die. However, this immune activation was much more modest in mice subjected to FOXP3 protein degradation. These are adult mice. And indeed, when we looked at the extent of immune activation, you can see that there is some increase in immune tone upon FOXP3 degradation, but it doesn't come close to the massive immune activation observed upon ablation of regulatory T cells. So this increase in immune tone largely persisted when FOXP3 degradation was extended to day 28, to a month. While by that time, all the mice subjected to ablation of Treg cells were dead. So this observation suggested that degradation of FOXP3 protein in differentiated Treg cells in adult mice doesn't impair suppressor function. And indeed, this is in vitro suppression assay that showed that the cells are indistinguishable in the cells that subjected to degradation of FOXP3, shown here in yellowish orange color, and control cells with intact FOXP3 shown here in green. So this observation raises the question whether FOXP3, once it's established transcriptional program, is capable of regulating gene expression, and specifically whether FOXP3 is capable of regulating its direct targets or its indirect targets in differentiated Treg cells. So this experiment is summarized on this plot where the log count, so this is RNA-seq analysis, and it was done by in bulk version of RNA-seq analysis and single cell version of RNA-seq analysis at different time points. So what you're looking at here in this column is count of gene expression of FOXP3 intact expressing cells and those that subjected to FOXP3 degradation. And you can see that largely gene expression is maintained, but some genes, few genes, are affected upon FOXP3 degradation in both resting and activated Treg cells. However, if you compare this column to this column, where we now compare gene expression between regulatory T cell wannabes I told you about that express reporter null allele and Treg cells that express functional reporter allele, you can see that there are many more genes that are FOXP3 dependent. So in this differential expression is summarized in this bar graph, where you can see that there are many more FOXP3 dependent genes than genes that are affected by FOXP3 degradation. So this is essentially genetic deficiency in FOXP3, and this is induced loss of FOXP3 protein expression. So importantly, the data that I don't show, and this is cut and run data for FOXP3 when we were looking at the binding of FOXP3 to its sites. We found that these genes, the few genes that are affected by FOXP3 degradation, are highly enriched, are essentially most of them are bound by FOXP3, whereas there was no enrichment in FOXP3 binding to genes that were FOXP3 dependent and differentially expressed in regulatory T cell wannabes versus functional Treg cells. And that was because we found before that the majority of FOXP3 dependent gene expression program is controlled by FOXP3 in indirect manner. So this data suggested that directly controlled genes by FOXP3 are still controlled by FOXP3, but indirectly controlled genes maintain expression after FOXP3 degradation. So this was loss of function experiment, and they thought to perform gain of function experiment. And for this experiment, she decided to use this FOXP3 LSL allele that I described to you, namely this allele where she can restore in a timed manner expression of FOXP3 upon administration of 4-hydroxytamoxifen. For this experiment, she has used heterozygote female mice that would harbor wild-type FOXP3 allele and LSL allele. So these mice are healthy, and control mice would be able to undergo recombination, would not be able to undergo recombination because there is no CRE-R, and experimental mice would upon activation of CRE-R encoded by CD4 locus. LSL will be excised and FOXP3 will be installed. And then this cells will be analyzed by RNA sequencing by sorting cells based on the markers that I described at different time points. So, and satisfactorily, what they found was that genes that were downregulated upon FOXP3 degradation were upregulated upon installation of FOXP3, and genes that were upregulated upon FOXP3 degradation were downregulated upon installation of FOXP3. So essentially, this complementary orthogonal experiment showed that we are looking at true regulation of FOXP3 genes, and that these genes are directly regulated by FOXP3. Another feature that I would like to draw your attention to is the very long time scale that this program is established. We found that it takes 2 to 3 weeks for the cells that subjected to installation of FOXP3 gene to reach the state of normally differentiating Treg cells. So this observation now suggested that perhaps the resilience of FOXP3-dependent transcriptional program that we observed in adult mice may not be held true or may not apply to neonates, to young mice, because in neonates, most of the Treg cells, the vast majority of Treg cells, are mesenchymic immigrants that spend too little time, and suggesting to us that perhaps in young mice, in the childhood, FOXP3-dependent gene expression program is vulnerable to the loss of FOXP3. So we tested together with Zhongmin this idea, and with graduate student Georgi Bilashvili in the lab. And a comparison of degradation of FOXP3 in adult mice and neonates mice showed that a loss of FOXP3 protein expression in young mice results in severe disease and phenotype that was indistinguishable from the mice that lack FOXP3 gene expression altogether, and that the loss of FOXP3 in Treg cells that in young mice resulted in loss of function based on in vitro suppression assay, and that also resulted in tissue inflammation. So this data suggested that there is a context-dependent differential requirement for FOXP3 for transcriptional and functional program of Treg cells, and unexpectedly long time scale of establishment of FOXP3-dependent transcriptional program, and that initial vulnerable to FOXP3 loss Treg transcriptional and functional program is capable of, as programs assume, a resilient state. All right, now I would like to switch gears and talk about a function of Treg cells. So early work from the lab showed that after differentiation, this TCR-dependent differentiation of Treg cells does not, so it's still differentiated Treg cells in a TCR-dependent manner still require TCR signaling for their function. Work from Andrew Levine in the lab showed that differentiated Treg cells require TCR signaling to maintain their function and to become effector function competent cells. Additional work from the lab showed that there are also TCR-independent function of Treg cells, and this is function and tissue repair and maintenance work by Nick Arpaia showed that production of tissue repair factor amphiregulin by Treg cells is independent of T cell receptor, and instead it's revoked by alarmin interleukin 33 and pro-inflammatory cytokine member of IL-1 family of cytokines, interleukin 18. So we have been interested in exploring further functionality of Treg cells in non-lymphoid tissues and their interactions with lymphoid and non-lymphoid hematopoietic and non-hematopoietic cells. So in order to address this question, Alejandra Mendoza, former postdoctoral fellow in the lab, started looking at neighbors of Treg cells in non-lymphoid tissues, and she noticed that Treg cells in non-lymphoid barrier tissues are frequently localized near sensory neurons, near axons of sensory neurons, as shown here in the staining, when CD31 outlines endothelial cells and tubulin beta-3 staining shows neurons, and FOXP3 staining shows Treg cells. And by looking at the distance, she found that they are in close proximity to sensory neurons, as shown here by the distance measurements. Considering that, and these tissues included intestine, lung, and skin. And considering that this sort of crowded space in the intestine, Alejandra decided to focus attention on the skin, considering dense innervation of skin by sensory neurons, as well as the lung and the intestine. So in the first question that she wanted to address, whether regulatory T cells since they're close to nociceptive neurons, sensory neurons, can affect nociceptor signaling. So she first used FOXP3 DTR mice I described to you, and instead of killing the cells, she decided to give them diphtheria toxin for 18 hours. As perhaps you know, diphtheria toxin works by blocking protein synthesis, and that eventually causes cell death. And the reasoning here was that if we give diphtheria toxin for short period of time, instead of killing cells, we will block protein synthesis in the cells and inactivate them. So the idea was to inactivate Treg cells and to look at the consequence at functional inactivation of Treg cells. So this is the control that shows that indeed administration of diphtheria toxin to these mice doesn't deplete Treg cells in the skin that are actually Treg cells in the skin are highly enriched. About 50% of CD4 cells in the skin express FOXP3 and are regulatory T cells, although there was some depletion after 18 hours in the secondary lymphoid organs, but no immune activation whatsoever. So, however, when Ali looked at IL-10 production, that has non-redundant function and suppressor capacity of Treg cells, she showed expected complete block in IL-10 expression by these cells due to blocked protein synthesis, whereas the cell numbers and immune activation was not affected in both lymph nodes and skin at this time point. So next, Ali decided to look whether this inactivation of Treg cells within 18 hours of treatment with diphtheria toxin would affect their activation and response activation of nociceptive neurons, sensory neurons upon administration of capsaicin. Capsaicin is a strong agonist for TRP1 ion channel, and that would activate and highly potent and very strong activation of nociceptive neurons. This was done using skin on the ear of the mice that are innervated by neurons residing in dorsal root ganglia, and activation of neurons can be measured by looking at CFOS expression using reporter staining by CFOS in sensory neurons. And also Ali performed tracing of innervating retrograde tracing of innervating neurons of ear skin and confirmed that they are innervating ear skin. So what she found then that inactivation of Treg cells resulted in significant increase in activity of sensory neurons in response to capsaicin, raising the question whether whether other forms of pain are also subject to this regulation. So she looked at hot plate sensitivity, which is thermal induced pain, and she observed that in this assay, when mice withdraw paw from hot plate and shorter time for withdrawal indicate higher sensitivity, so decreased latency of response to hot plate indicates higher activity of sensory neurons. Cold sensitivity was also affected. This is by applying acetone on the paws of mice, and mechanical sensitivity on von Frey test was also affected. So, of course, the next question we ask whether increased sensitivity to noxious stimuli can lead to exacerbated tissue inflammation, and this question is based on earlier work by Dan Kaplan and Uli von Adrian's lab, who showed that so-called sensory neuron activation arc, that sensory neuron activation triggers inflammatory reflex and exacerbates inflammation in the skin upon application of imiquimod. Imiquimod, as I'm sure some of you know, is a ligand for TLR7 toll-like receptor in mice and in humans. So what then Ali found that depletion of regulatory T cells, in this case, results in significant augmentation of activation of sensory neurons measured by CFOS expression in response to imiquimod. as well as increased production of pro-inflammatory cytokine IL-23 within the short time window. This was our preferred readout for increased activation of innate immune cells, and IL-23 is known to be produced in this model by dendritic cells sensing imiquimod. So this increased sensitivity, increased inflammatory responses observed upon depletion of Treg cells in imiquimod challenged mice was absolutely dependent on sensory innervation, because chemical denervation of sensory neurons upon administration of resinoferatoxin completely erased the observed differences. So then, of course, the question was, what is the molecular mechanism and cellular mechanism of the observed effect? And we started looking at potential candidate, and our primary candidate was proenkephalin. We found that a subset of skin Treg cells express proenkephalin. Proenkephalin is a precursor for enkephalins, endogenous opioid neuropeptides that inhibit pain perceptions through binding to opioid receptors. There are 3 of them, and that PENG-deficient mice lacking PENG gene in the germline have increased sensitivity to pain, develop anxiety and aggressive behavior. So through generation of a reporter mouse, Alejandra was able to demonstrate that skin is the site where a great number, 50% of Treg cells residing in the skin are expressing or have history of proenkephalin expression, and the same was observed in the lung, and some increase in the lung was minor increase, but these are in steady state. The majority of Treg cells present in the lung are in the vasculature, not tissue resident in healthy mice, but also in the large intestine, whereas proenkephalin expression by Treg cells in lymphoid tissue, second lymphoid organs, was not observed. So by using genetic trick, by generation of mice that allowed Alejandra to selectively ablate PENG-expressing Treg cells, and to save time, I'm not going into the technical details how these mice were generated. The bottom line is that Ali was able to deplete or inactivate PENG-expressing Treg cells. And this is the experiment when Treg cells were now depleted, PENG-expressing Treg cells. There was no change in Treg cell frequency in second lymphoid organs. There was a decrease in PENG-expressing Treg cells in the skin, as expected, a decrease. And when the cells were inactivated for 18 hours, similar to what I showed you before, not depleted but inactivated, they showed decreased latency of thermal response, and they showed increased CFOS activation, and they showed increased IL-23 production upon imiquimod painting. And in this case, this is a depletion of Treg cells for 40 hours. So what about PENG non-expressing Treg cells? And another genetic model generated in the lab that Ali has used allowed her to selectively ablate PENG non-expressing Treg cells and to leave only PENG-expressing ones in mice. And she found that this results in decrease in Treg cells in second lymphoid organs and in the skin. However, this decrease was not associated with changes in thermal response in 18 hours. So inactivation of the cells didn't change thermal response, and it didn't change CFOS activation, and it didn't change IL-23 production, innate immune response. So suggesting that a population of PENG-expressing Treg cells is required to restrain sensory neuron activation and associated tissue inflammation, and that PENG is a mark of this neuromodulatory Treg cells. So, of course, then obvious question is whether enkephalins themselves are required for Treg cells to modulate nociception and inflammation. Of note that the Treg cells expressing proenkephalin also expressing convertase, the protease that cleaves precursor polypeptide into 5-mer enkephalin peptides that act on enkephalin receptors. So for that, allele-generated mice that harbor floxed allele of proenkephalin and then combine this allele with FOXP3 CRE that was allele generated long time ago. And then she acutely ablated proenkephalin in this mice upon administration of tamoxifen, let mice rest for 7 days, and then applied imiquimod to this mice. And she also ran the pain sensitivity assays. As shown here, this ablation of pink in mice didn't result in any measurable immune activation. It resulted in increased heat sensitivity and increased mechanical sensitivity. It didn't affect cold sensitivity, but it's very noisy assay, and we suspect that it's just noisy and insensitive assay for measuring of cold-induced pain. However, in imiquimod painting assay, Ali observed significantly increased FOS activation and increased IL-23 production in mice where proenkephalin expression was ablated in Treg cells. And again, ablation of sensory neurons erased all the differences observed. So to look at the long-term consequences of PENG deficiency in Treg cells on skin inflammation, Ali extended the imiquimod treatment to more long-term treatment and looked at pathological sequelae and found that both frequencies of IL-17 production producing gamma delta cells, neutrophil numbers in the skin of mice harboring PENG deficient Treg cells were markedly decreased, and then skin thickness and skin inflammation was significantly increased. So how PENG expression is regulated in Treg cells? So Ali found that PENG expressing Treg cells, and that was done using plate-seq-based SMART-seq2 single-cell RNA sequencing, then PENG expressing Treg cells are enriched for Treg TCR signaling. Associated gene expression represent terminally differentiated cell state, as shown by TCR up signaling, and that she also found that loss of TCR in Treg cells resulted in a loss of PENG expression. However, Ali found that activation of T cell receptor in PENG non-expressing Treg cells does not upregulate PENG expression. So that raised the question of what signals are required to induce PENG expression in Treg cells. T cell receptor signal is required, but clearly is not sufficient. Examination of proenkephalin gene promoter showed a prominent presence of motifs for a glucocorticoid receptor. As shown here, it's NR3C1 is a gene name for glucocorticoid receptor, suggesting that glucocorticoid receptor signaling can facilitate proenkephalin expression in Treg cells. And indeed, application in the presence of dexamethasone, there was a significantly increased expression of proenkephalin in Treg cells in vitro, increased production of metenkephalin peptide measured by ELISA, and that ablation of glucocorticoid receptor in Treg cells resulted in a markedly decreased expression of PENG gene. Together, this data suggested that a neuromodulatory function of enkephalin-producing Treg cells is capable of directly modulating activity of nociceptive sensory neurons and restrain tissue inflammation through production of endogenous opioid neuropeptides. So I will just highlight a few slides on the role of regulatory T cells in cancer. Treg cells, as we have shown and others have shown, are prominently present in tumors. This is analysis of breast cancer patient tissue samples. These are tumors. These are normal breast tissue isolated from counterlateral breastectomy from the same patients, and this peripheral blood mononuclear cells. And you can see that in the tumors, Treg cell frequency is significantly enriched in comparison to normal tissue and comparison to peripheral blood. More aggressive types of breast cancer, such as triple negative breast cancer, are more enriched for Treg cells, and also tumors, higher grade tumors, are also more enriched for Treg cells, suggesting that perhaps Treg cells can be used as a target of cancer and cancer therapy. And that was observed again by multiple laboratories. Paula Bose, together with George Plitas in the lab, showed that acute depletion of Treg cells in FOXP3-TR mice results in profound restraint, a punctual depletion of Treg cells, restraint of the tumor growth in this mice, both primary tumor and metastatic tumor. And this was tumor that was resistant to PD-1 blockade and in a genetic model of breast cancer. So with this in mind, we put efforts and attempts to identify potential target for somewhat selective, for ideally selective depletion of regulatory T cells in human cancer. And George Plitas and Kasia Konopatsky published this paper now 10 years ago, back to back with a similar study from Massimo Pagani's lab from Milan. And in this work, George and Kasia found that intratumoral Treg cells across different types of human cancer are highly enriched for expression of chemokine receptor CCR8, as shown here. And this is expression in tumor, in normal breast tissue, in the blood, and these are other types of cancer are shown. So this observation led to massive efforts to develop targeted therapy. We do have conflict. We do have in IP for therapeutic CCR-targeted Treg cell depletion and develop our own panel of CCR8 depleting monoclonal antibodies. Currently, there are 12 clinical trials conducted, including the panel that we have generated, licensed to Takeda. And currently, there are phase 1, 2, and 3 clinical trials across different types of cancer focused on depletion of Treg cells based on CCR8, using CCR8 targeting antibodies in combination with PD-1 and other combinations. So we had also a question whether there are other strategies of targeting regulatory T cells, and we're thinking of this observation that I shared with you of FOXP3 protein expression, vulnerability Treg cells, and resilience of Treg cells to FOXP3 protein loss. And in this case, we thought to investigate whether intratumoral Treg cells are assumed vulnerable state and can be much more sensitive to the loss of FOXP3 expression than the ones that are not tumor resident, because in the tumors, cells divide robustly and also are exposed to drastically different metabolic environment and also deprived of interleukin-2. So in this experiment, using this FOXP3 ID allele that I described to you, chemogenetic tool, FOXP3 ID mouse that were transplanted with B16 ovalbumin expressing melanoma cells. Cells were let grow, and then mice were treated with 5-phenyl iodoacetic acid to degrade FOXP3. And that, unlike the gene expression, minor gene expression changes that I showed you before, resulted in profound changes in gene expression. So this is the UMAP of single-cell RNA-seq data from mice, from cells harboring intact FOXP3 allele versus subjected to FOXP3 degradation. And you can see that the cells are not mixed. And we observed that about 200 to 500 differentially expressed genes in 4 major clusters that encompass all major flavors of intratumoral Treg cells. And this depletion resulted in significant restraint of tumor progression. Tumors were rapidly shrinking, and that was not associated with any significant adverse effects. Mice didn't lose weight. They were not really dying. If anything, that increase in immune tone elsewhere in non-draining lymph nodes, secondary lymphoid organs, and non-lymphoid tissues was even less pronounced than in intact mice not harboring tumors. So therefore, FOXP3 protein degradation does restrain tumor growth in the absence of frank adverse effects and represent potential novel means of intervention in cancer. So with this, I would like to close by acknowledging people who did the work. Wei Hu with degradation, with Eric and Georgi helped her, graduate students in the lab. Alejandra, together with Stas, Paul, and Regina, graduate students in the lab, are responsible for the work focused on role of enkephalin expressing Treg cells in suppressing nociception. And I highlight George and Kasha's work and Paolo Bo's work. Gabe Dolstein helped Wei analyzing gene expression. It was gene expression. Gabe was graduate student in Yuri Pritikin's lab, our long-term collaborator. Chitra Dahi and her postdoctoral fellow Danila Correa helped with pain assays, along with Susanna Manenti and Sebastian Jara, who are graduate students in Alejandra's lab at Scripps. Alejandra runs her own lab at Scripps, and Wei runs her own lab at Yale. And Sneha Mitra and Xiao's work I was unable to fit into 1-hour lecture. This is it. Thank you, and I'll be happy to answer questions if there are any.
Fantastic, Sasha. Thank you so much. We are open for questions. Normally, we take questions from the students first, and then from the PIs. Anybody here? Anybody at your side, Bruno? Students?
No, not students. Not students.
Not students.
We do have
okay.
questions here.
Yeah, okay. So I have multiple questions, Sasha. It's brilliant work. I will start with one specific and then a more general questions. Regarding your PANC non-expressing Tregs depletion, you mentioned that I was amazed that there was no compensation for another Tregs. So is there like compartments of Tregs that are independent between the PANC expressing and non-expressing cells?
Right. So we think that the PANC expressing cells are the ones that are activated and are exposed to glucocorticoid signaling, something that for the lack of time I didn't explain well, but the reason for enrichment, we believe that the reason for enrichment of proenkephalin expressing Treg cells in the skin is because the skin is the major site of local glucocorticoid production. So keratinocytes produce glucocorticoids. They're equipped with enzymes involved in biosynthesis of glucocorticoids, and deficiency in glucocorticoid production in the skin in mice and men results in spontaneous inflammatory disease. So glucocorticoid expression in the skin will be activated in response to inflammation, injury, and infection, and that would induce expression of proenkephalin in skin-resident Treg cells. Intestine also is a site less prominent than the skin of local glucocorticoid production at the steady state. So we analyzed data that I showed you. This was analysis of mice at steady state. Upon challenge, there is further increase in proenkephalin expression in the skin. So in response to you, we looked at the T cell receptor usage by proenkephalin expressing and non-expressing cells. It's because we had unrestricted repertoire, it was not particularly informative. We couldn't say really. For sure, both express fairly diverse receptors, but we cannot say whether the repertoire are distinct or not because there was no major expansions, clonal expansions between proenkephalin expressing and non-expressing cells. I assume that activation of T cell receptor, glucocorticoid receptor, in proenkephalin non-expressing Treg cells, as we shown in vitro, would result in vivo also in expression of proenkephalin. But since we are depleting proenkephalin expressing Treg cells, and even if proenkephalin is upregulated in proenkephalin non-expressing Treg cells, they will be depleted. They will be gone. So I think that proenkephalin is active. Likely there will be other effector mechanisms contributing to activity, but clearly proenkephalin expression and production of opioids and enkephalin opioids has non-redundant function in toning down nociception. Something that I didn't mention in this regard, if we extend observation of mice without challenge, when we ablate proenkephalin expression in Treg cells and look after 4 weeks, we see that the basal level of activation of nociceptors in dorsal root ganglia is increased. So the basal tone of nociceptive activity is increased.
It's incredible. I'll allow everybody, somebody else to ask, and then if we still have time, I'll ask another general question. Bruno.
Hi. Actually, I have a question, and Mira has one too. But it's actually a follow-up on your question, Gustavo. Is that if there is any room for the cell types in this interaction between Tregs and the neurons, particularly room for mast cells that are known to participate in this neuroinflammatory loop induced by capsaicin, for example. So, did you guys ever checked for the cell types?
Yeah. It's a very good question, and of course that we were thinking of intermediaries in a way, and there is something that we are so that another line of studies is related to control of mast cells by extratymically generated Treg cells that we have observed. And so far we looked at that. We tried to look at the direct contacts. We didn't observe really strong evidence. We also tried to use the tool that Gabriel Victor developed, right? So lipstick Treg cell that express lipstick tool that would allow for tagging of directly contacting cells with Treg cells, while we able to tag, say, macrophages, dendritic cells, we were unable to tag mast cells. There must be connection. We reported massive expansion of mast cells, mastocytosis in the intestine, but also some in the skin in mice where we genetically impair extratymic differentiation of Treg cells. In this case, we don't know. We don't think that. We think in this case, we think that it's likely direct. We believe that it's direct activity of enkephalin acting on enkephalin receptors on sensory neurons. I can't hear you.
Sorry, battery problems. Is it okay now?
Yes.
Okay. So thank you very much for your talks. Really beautiful data. I have a couple of questions. One of them is about the origin of the PENK expressing Tregs. Do you think they could be from the natural Tregs, or they will be from the differentiating ones in the skin?
Yeah, I think that it's a good question. We think just percentage-wise, a good proportion of them is likely of thymic origin. And a good proportion of the cells are actually the ones that seed skin in early life.
Mhm.
So there is another study that we are currently trying to finish that suggests that. So I think numerically extrathymic Treg cells contribute to relatively minor proportion of skin resident Treg cells. The majority of them are, we believe, early life Treg cells that are generated and getting out in the periphery in neonates and young mice.
And the second question is about the applicability in disease. Do you see any different cells in psoriasis patients versus melanoma patients? Do you think that could be more, it could add up to the pathologies?
Yeah, it's a good question. We have not looked. We didn't have access to psoriasis patient samples, although I think we may look at the published single cell data sets. But ideally would be to not rely solely on mRNA expression. In the tumors, there are proenkephalin expressing cells, and also in mice in the studies, there is another subset of hematopoietic cells that express proenkephalin, are gamma delta cells, and Ali in her lab is exploring potentially communication of gamma delta cells, perhaps with other types of neurons and non-neuronal cells, but they also express proenkephalin.
Thank you very much.
Alpha beta cells don't.
They don't?
Non-Treg cells do not express.
Okay.
We have a question from Leandro online. Leandro.
Hi, Sasha. Very interesting data. I just was curious about these resident Tregs, if they can produce like neuropeptides or growth factors that could sustain these neurons.
We know that they do not produce BDNF, right? If to think of this type of growth factors. Treg cells do produce other factors. So I mentioned amphiregulin that has been implicated in tissue repair, and we showed that it's produced by Treg cells in TCR-independent manner. So essentially, that cells need to be activated through T cell receptor in order to migrate into the tissue, but amphiregulin expression and production will be comparable in cells lacking T cell receptor. So amphiregulin, it does work as a neuronal repair factor in addition to epithelial repair factor, and it has been shown to in traumatic brain injury model by Aki Ishimura to support repair in the brain in mouse model of traumatic brain injury. I'm aware of at least one company, cell therapy Treg company, developing off-the-shelf Treg cells for these indications to support neuronal repair and injury. So, and then there are other data. In this case, it would be immunomodulatory factor, and that was published by a very recent paper by Diane Matis-Christophe-Benoit's lab, suggesting that Treg cells, through production of interleukin-10, can support neuronal stem cells. So that was recently reported, I think, in Science Immunology.
Sasha, we have a question.
You know, some days that they can produce like catecholamines or like neuropeptides like
Well, there
neuropeptide
are,
age
so
or
Takmak has the data. So it's acetylcholine esterase expressing T cells. He published a bunch of papers on that. Treg cells are actually prominent expressors, but also non-Treg cells, effector cells can do that. And we have another study on poorly understood neuropeptide that it's currently under revision. It's too early to tell.
Okay, yeah, thank you. Very interesting.
Sasha, there is a question in the chat by Julio Sharfstein. Julio, are you around? Can you open your mic and
maybe you can read if you see it in the chat.
Yeah. I'll just check whether Julio is around and he can ask himself.
I just
Okay,
don't
can
think
you
that.
hear me? Okay.
Yeah.
Yes, Julio, go ahead.
Okay, hi. Yeah, the question, well, it's a fantastic talk. Of course, there must be some dependence to generate the mature enkephalin ligand for these effects. And I would assume that probably there are so many cells around that can express the key convertases. But of course, you might know also if there is transcription of the convertases by the differentiated Tregs themselves, if they have autonomy to do it.
Yeah, Julio, I mentioned this, maybe I just was in passing. The cells express convertase.
Okay.
They express actually two convertases for proenkephalin and pretty high level. So, and they generate enkephalins based on ELISA. So we can show that reviewers asked us to show that they can generate.
That's very important. So they have autonomy to do.
Yes, that's right.
The effector function. Yeah,
exactly.
Fantastic.
Exactly.
Fantastic. Thank you so much.
Yeah, you're welcome.
Sasha, just one more question. How this PANC non-expressing versus expressing, the inducible versus natural Tregs, the ones that depend on the TCR signaling and the ones that do not depend, contributes to inflammaging?
Well, I don't know how they contribute to inflammaging.
So this,
I think that the sensory pain would result in increased inflammatory tone for sure. So I think that there is seemingly non-redundant function of proenkephalin, at least in mice, of proenkephalin production by Treg cells. Other cells also produce proenkephalin. But I think I think that the overall generalizable message is that although multiple cell types can produce particular gene product, there is a cell type specific function. Frequently, it's probably more frequently than not, it can be non-redundant function because cells would form specific circuits that can be affected by cell type specific protection of mediator shared by more than one cell type. Proenkephalin is just one example, but there are other examples. But yeah, we don't know about that.
right, but are there any of these populations that decreases in activity or in size during aging or
we have not looked at it. Yeah, so we are trying to keep our mouse colony
short term.
Yeah, so it's large and we're trying not let mice sit too long. Yeah, I think that aging experiments are expensive.
Okay. All right, Sasha. I think we don't want to take more time of you. You mentioned that you need to leave. Thank you so much. It was really a pleasure and honor to have you among us today, and really hope to see you soon here in Brazil or any part of the world.
Thank you. It was a pleasure.
Thank you. Thank you, Sasha. Thanks a lot.